Welcome to the 2021 celebration presentation on bioactive algal metabolites in Ambystoma maculatum and Ambystoma mexicanum tail regeneration. My name is Matt Cherubino, and together with my team of fellow undergraduates, we have been conducting an ongoing investigation over the past 2 years. For introductions, I am a biochemistry and molecular biology major who got interested in regeneration science my freshman year in Dr. Kearney's lab. Preston Southwick is a junior health science major and biology minor. Christine Sidamic is a junior biology major and chemistry minor. Nathan Motel is a sophomore biology major. Sarah Feldman is a junior biology major and religious studies minor. And Zoe Bender is a junior biology major and chemistry minor. Our first species of interest is the yellow spotted salamander, Ambystoma maculatum. Christine named her Mary back in March, and on the left you can see her egg clutch, which she laid in captivity. Our second species of interest is the axolotl, Ambystoma mexicanum, and it is critically endangered in Mexico, however widely used in laboratories around the world. On the left is an embryo inside a jellycoat or egg capsule without algae, and on the right with algae. The algae is Oophila amblystomatis. It is the symbiont of interest in our study. And this picture shows an 8x7 grid that represents one condition of an experiment, in this case, the with algae light condition. Each row represents a new embryo, and each column a new time point, such that as you move from left to right, you're looking at time increasing in DPA, or days post-amputation. Now, each of these can be measured in ImageJ, and the measurements can be compiled and processed. This is a screenshot of an RStudio code, the 4 lines of code to most of the statistical heavy lifting, and the last line plots a Tukey plot with a 95% family-wise confidence interval. These statistical tests can give us graphs like this, which show average regrowth and days post-amputation. We are able to find significant differences, in this case, between the algae light group and no algae dark group for Ambystoma maculatum. Now, both of the conditions were force hatched from their egg capsules during the rearing period prior to amputation. So the algae light group is able to regenerate faster. And this is the really interesting finding that most of the ongoing research is focused on in our laboratory. So for measurements, this shows a total length or TL measurements, and I highlight the cloacal opening as well. So it was an older method of measurement used in the literature and by us to figure out if there were differences in the regrowth rate. However, over the past many experiments, we've now just finished experiment 13, we've developed something called the cloaca correction, which is more accurate than total length because it eliminates regrowth that occurs, or rather just growth that occurs from the tip of the head to the cloacal opening. Now, there are more accurate methods in the literature, for example, measuring by the amputation plane. However, this is difficult to do, and so the cloaca correction is a good, reasonable compromise between accuracy and difficulty. Here we have representative 20x magnification images at 0, 3, and 7 dPA. You can see how the tail tip regenerates 0 dPA immediately after regeneration to 3 dPA, where there is a tissue remodeling followed by notochord extension, and then by 7 dPA you have the tail tip, the tail fin completely surrounding that extended notochord. Now for the future directions, we have 4 main directions that the project is going. It is an ongoing project, and the first of them is the use of an alexafluorazide. This is Clickit EDU chemistry dye, and it will incorporate into DNA and S phase of mitosis. For those of you familiar with bird U labeling, it's very similar. It's comparable, except that the alexafluorazide is really just an improvement on the now outdated precursor. We're also working on a column, a deoxygenation column, This was a schematic I designed, which shows water being deoxygenated through the column and then reoxygenated through an aeration ladder to a specific concentration, a wanted concentration that we can rear the embryos in. There is a known oxygen exchange that occurs between the symbiont, the algae, and the embryos. And so by deoxygenating and then reoxygenating selectively the water, we're able to figure out not only if that exchange is responsible for observed regeneration rate differences, but also we will be able to figure out how much of that difference is attributable to the oxygen exchange. And the other two directions, the first, University of North Carolina Wilmington, which sent us extracts previously, which covered many experiments to figure out if we could find a chemical extract fraction of the algae that promotes that regeneration. They now have refined extracts they sent us, and we are currently performing the screen on those refined extracts. Second, the vast new direction, Ellie Vickers, a previous Gettysburg graduate, and Ryan Kearney's lab class will be performing and are performing a bacterial screen to see if any of the bacteria found around this natural algal and embryo symbiosis, salamander symbiosis, if they have any effect as well on regeneration rates. So in either case, we can apply the deoxygenation column and this molecular metric for tracking regeneration, the clicket chemistry, EDU cell labeling, which again, that we can use fluorescence to figure out where the replication is occurring in mitosis. We'll be able to confirm and better look at these two different types of experiments we're running. And so finally, I'll leave you with my team. I'm incredibly proud of the work we've done so far and the direction we're headed. Thank you guys so much. And lastly, I'd like to thank Dr. Kearney, the research mentor and sponsor, for allowing us to do this research in his laboratory. Thank you.